metabolism, translation, splicing of the genetic material evolved around RNA, and RNA used to act as a genetic material as well as catalyst. Okay, it can act as a catalyst, shows the catalytic reactions, biochemical reactions in living systems that are catalyzed by RNA. It can act as a genetic material, hence, but it is unstable due to its molecular structure. Therefore, DNA has evolved from RNA. DNA has evolved from the RNA, and with chemical modifications that make it more stable. DNA being a double-stranded structure and having complementary strand further resist, it has complementary strands, okay, further resist changes by evolving a process of repair. Now, what do you mean by replication? Okay, so the proposition that double helical structure of the DNA, it has a double helical structure, by Watson and Crick had immediately proposed that a replication, what is that? A replication of the DNA. Okay, here replication, how it occurs? It has not escaped. Okay, and as per our notice, the specific pairing have postulated specific base pairing. Okay, immediately suggests that a possible copying mechanism for the genetic material. Now, the scheme suggests that two strands of would be act as a template. Two strands of DNA act as a template for the synthesis of new strands or complementary strands. What is complementary? Complementary is nothing but new strands, formation of new strands. If you take the DNA as a double helical structure, you see here, okay, the two strands here can act as a complementary strands for the synthesis of or for the formation of complementary strands or new strands of DNA. After the completion of replication, each DNA, if you see, each DNA molecule would have one parental, one newly synthesized strands. Suppose this is a strand, okay, it forms a new strand or complementary strand like this. This is another strand, it can form a new strand or a complementary strand like this. Okay, each strand will have this is the parent strand, okay, parent or parental complementary DNA strand. Okay, and this scheme was termed as a semi-conservative. Okay, the process of formation of these complementary DNA strands is called semi-conservative DNA replication. What is that called? Semi-conservative DNA replication. Finally, I would like to say that what is the experimental proof for this? Okay, if you take the experimental proof, it is now proven that DNA replicates semi-conservatively. There was an experiment. First in E. coli bacteria, it was performed by and after that it was performed in higher organisms such as plants and human cells. Matthew Messelson and Franklin Staple performed this following experiment in 1958. What they did there? In the first step, they have grown E. coli bacteria in the medium of containing okay, NH4Cl, okay, isotope 15 NH4Cl, okay, 15th N refers to nitrogen, okay, 15th nitrogen isotope. What is that? 15th nitrogen heavy isotope as the only nitrogen source for many generations. As a result was that 15th generation nitrogen isotope was incorporated into the newly synthesized DNA as well as. Okay.